This is the prep for lecture 8. And like the P element one, I'm going to mostly go over the whole lecture in advance because I think for most people they will need to think it through many times, 3 or 4 maybe, before they really feel comfortable with it. And so if I do 2 of those repeats, that means there's only a couple more you need to do on your own. Okay, and the reason why I'm being so trying to be more detailed than some of the other preps is that this is the subject of a real research project, and you are going to write your first proposal or paper, that is, which is a proposal about this topic. And so I have posted, I haven't yet, but I want to make sure they're up to date and maybe edit and clarify some of the documents before I completely have them posted. But I will have a whole module filled with, there will be some guidance on how we're grading, what we're looking for, and how to structure it. There's a very specific structure for a research proposal. I've listed topics in a particular order, which I think is worthwhile considering writing in that order because that makes logical sense. Here's the gene, here's why it's interesting, this is what we know, this is what we don't know, we want to find out, here's why we're using flies, and this is what we're going to do. That tells a little story, and that's what I think is really useful. Okay, and on the screen, I have screenshots on the front here of a few pictures from Dr. Sandra Schultz's blog post, because she's got a blog where she describes for the public some of the research that's going on in her lab, and she does quite a variety of things. So this is just one of the projects. And so when you first arrive, you will find this, and this basically is the project that we have been working on together, Sandra and I, since about 2011, 2012, in there somewhere, and it just explains sort of the background. And then what I would suggest, you don't have to go through all of these, but so if you start here, then the first thing is to this is our research question. This is what we want to know. This is a screen that my class did for a few years between 2011 and 2016 or 17. And then some other things. If you're interested in epigenetics, then position effect variegation might also interest you. But for our purposes, these are the posts. Now, Sandra's writing style is very ornate. She tends to write in fairly complicated sentences, but you still can usually glean some good information. She's 1,000 times smarter than I am, for example. And so you can really tell from her writing that she knows a lot, and she's got like an extensive vocabulary and so on. But I still think, there are images that show how we did our original screen and so on. I don't think you necessarily need to look at these ones. You might find this one of particular interest and so on. In any case, this is one of the resources that is available for you. I've also posted some, and I found a really interesting recent paper too, which I'm going to post. So I'm going to post a few reference articles. You don't have to read all of them, but I would suggest reading a few of them, maybe 3. And more if you have the time and interest, but I recognize not everyone is going to have enough time to do, read 10 papers on the subject. If you haven't read a paper before, I plan to post a little bit of guidance on how to approach reading a scientific paper. They have a particular structure, and depending on your familiarity with the topic, there are different ways of going about it, and I'm going to make some suggestions on that. Okay, I think that's good. And so basically the overall goal, so here's our project. We want to make an insertion element, P element mutation in the DMAP1 gene in flies, fruit fly. And the method we plan to use is referred to as a local hop. And what that means is that we have the DMAP1 gene sitting on chromosome 2, and there's a P element in the strain that we're going to use. There's a P element right next door, and we're just going to try to move it into the gene. And it's called a local hop because what it is, it's the P element moves in a very short distance to a nearby gene. So it ends up close to where it started. P elements move all over the place. They can move to other chromosomes. They can move hundreds of thousands of kilobases away from where they are originally. But overall, if you look at research where they examined the mobilizations, they basically took an element, they mobilized it, and then they tried to find out where it had moved to. And on average, 1 to 2 kb from the original position was approximately where it was found. And that would be perfect for us because if it was 1,000 kilobases away, the element would go right into that gene. Okay, and I'm going to talk about certain mutations. I don't think I have pictures for all of them in here, but if you go to this website here, it shows it's a German site, but it doesn't matter because you'll be able to see the names of the genes and then what the mutant phenotypes look like. Okay, and so from this lecture, I should have underlined this. I don't know why I didn't. Oops. I always, on the outcomes, try to underline the thing you are going to do that demonstrates your understanding. For purposes of being able to write your background nicely, we want you to just be able to describe the gene we're targeting just a little bit. It doesn't have to be every single detail about it, but why, the name of it, how it was discovered, and maybe a couple of interesting things about it based on the literature that you read. Properly referenced, I'll provide guidance on how to do that. As well as being able to outline the steps of the genetic screen that we're proposing and the reason for each step. I will tell you that I had directed studies do this screen a few years ago, 2018, 2019, and we do actually have a mutation. So you have the comfort maybe of knowing that what you're proposing was done and in fact didn't require nearly as much work as I was expecting. And all of these outcomes as well are going to help you write your paper, right? So being able to describe the gene and why it's interesting, being able to outline the steps of the screen that we're proposing and what each step is for, yes. Reading those 3 papers to get some more background understanding. And just look at the titles in the abstract. Some of them will seem more interesting to you than others. And so pick the ones that you think are the most interesting. That way you have a little bit of choice about reading a paper can be a lot of work, but at least you will be able to choose the ones that sound like they're more, close to your interests. And then writing your clear and well-organized research proposal. Okay, and so why are we interested in this gene? So Dr. Schultz and I did our PhDs in the same lab, and we were friends years before that. We first met when I was her genetics TA, when I was a master's student and she was an undergrad. Now she's a prof, and so we've maintained an interest in genetics along with a good friendship over the years. And she was basically on the committee of a master's student who was studying a gene in WASP, and so she was of course helping advise about the research, and it turned out that the gene was called DMAP1. So DNA, this is how we name it, DNA methyltransferase associated protein 1. So that's where the name comes from, and it was named this and identified because it was identified based on its protein binding to DNA methyltransferase 1, DNMT1. That's what this is. This is the human one, and I believe this is Amphioxus, which is it's sort of the precursor to the vertebrate line. Okay, oops. And the student was studying this in Nisonia, which is a parasitic wasp. which is very interesting to work with, but you have to grow them on rotting liver, which is a very unpleasant way of culturing your research organism. In any case, DNA methylases are very interesting because when DNA gets methylated, most of the time that's involved in repressing gene expression. So because if you add methyl group CH3 groups, that's what a methyl group is, and you add those to DNA, and suddenly the transcription factors that are supposed to be maybe expressing a gene, they can't find the DNA and bind the DNA sequence, because when you add those little methyl groups, you're changing the shape of the DNA in that position. And so that makes it not accessible. So not 100%. Occasionally there are certain kinds of methylation, not of DNA, but of the proteins associated with it that might be associated with activation. But generally methylation is involved in repressing genes. And if you think about how an embryo develops, as when the egg is first fertilized, there's a whole ton of genes. Some are only required to be expressed in the anterior part of the embryo, and some only in the posterior part. So in the posterior part, certain ones are going to be permanently shut off because you don't want those being expressed and messing up the way the organism is patterned, with the wrong structures in the wrong places and so on. And then in the anterior, all the specific genes for the posterior get shut off. And so that's one of the purposes of methylation. There are others, but that's one of the commonest ones. Okay. And so what do we know about it in mammals? Well, there's a few things. One is there are possible cancer connections. So one of the things we know about certain kinds of cancers is that there are certain genes that are shut off through methylation in cancers. And that means that the gene that is supposed to be expressing a protein that helps to check DNA for damage and prevent cells dividing if they're damaged — cancer is a DNA damaged kind of disease — then if you could reverse that methylation, you could, in theory, you could reverse the cancer by reactivating. There's nothing wrong with those genes, it's just that they've been turned off. The sequence is correct. So if you could take away the methylation, you could perhaps have them functioning as wild type and therefore curing or treating the cancer. Now, one of the things to be aware of for most of the, not all, but many of the mammalian experiments, they're done with cultured cells. And if you find ones that are done with cultured cells, you need to be aware. So if you look at papers and you go, this is really cool, one little caveat is that those cells, these are usually what we call immortalized cell lines. And they are basically slightly transformed towards being cancerous. They will not necessarily have the correct numbers of chromosomes and so on. A lot of, if cells can grow in culture without having a specified lifespan, without dying eventually and finishing the number of cell divisions they can do, they are not genetically normal, so they have had some genetic alteration. And what that means is that we always treat the culture work with a little bit of caution. Okay. Now there's some work that's been done in mice. You'll see in one of the paper that is seems to be related to stem cell maintenance in mouse embryos. So if you mutate DMAP1, it alters the ability of those stem cells to be maintained. Stem cells, as most of you probably know, are undifferentiated cells that can be used, for example, in healing, in production of gametes. In your body, you have stem cells in a number of your different tissues. So think about skin. You get a cut or some sort of damage. The stem cells in the skin will produce more cells that can be differentiated into skin and will help you with your healing process. There are even stem cells in the nervous system, and there are people that are working to try to activate those when, for example, there's a spinal cord injury, something like that. And so this was done in the living organisms, and you might find that really interesting. There's also one that I posted or will post very soon about planaria. When planaria are flatworms, they kind of look something like this. I think they're a bit sort of longer, and they've got a little stomach that you can actually see, and they have two sort of weird eye spots, and it makes they kind of look vaguely cross-eyed because the eye spots are not actual eyes the way we see them. But you know, you can, as some of you know, that you can cut a planarian in half, and both halves will generate a new organism, which is really an interesting feature. And we like studying organisms that can do regeneration because they can maybe help us when we have loss of limbs or loss of digits or other structures. If we could figure out how to harness that regeneration, it would be very helpful for us in terms of injury and so on. But if you decrease DMAP1 expression, you decrease the ability overall for this kind of regeneration. And there are a number of different processes involved in that that DMAP1 may be having an effect on. So we think a gene that does these kinds of things, that maintains stem cells and allows for proper cell division and so on, and is involved with regulating genes correctly, that should be an essential gene. So just based on the kind of gene it is, we think it probably is. But we're not yet 100% sure, and I'll explain why in a minute. And you may or may not agree with this, but Sandra and I decided that it'd be kind of cool to find out what we can find out about what it actually does in flies, because it hadn't at that time been studied at all in flies. There have been a couple of papers that have come out since, but they haven't really addressed this question directly. Okay, so why do we think that inactivating DMAP1 should be a lethal condition? That's what an essential gene means, required for life. So it's associated with a protein that it encodes a protein that most likely does something important involving gene regulation, right? So we can assume that would be. Now, Sandra in her lab did some work using a method we'll cover later. Some of you know what RNAi is already, and some of you have not learned about it. But a directed knockdown of the expression did lead to lethality of flies. So this is a way of reducing expression of a particular gene. However, there is a problem with this method, which is that sometimes, and we can't always detect this, it is sometimes possible that your method knocks down some other genes besides the one you're intending. So what that means is that there's a small, it's not a huge chance, but there's a small chance that the lethality that Sandra observed in DMAP1 knockdown flies is due to another gene besides the one intended. So it could either be totally another gene and not DMAP1 at all, or maybe it could be the combination of reducing DMAP1 and maybe by itself reducing the expression of that gene isn't going to cause lethality, but when you knock down 2 or 3 others at the same time, now that's too much of a problem for the organism and it can't survive, right? So this is the idea. So what we wanted to do is we wanted to directly inactivate the gene, okay? And we wanted to make a deletion. I'll explain really briefly how we did that in the next recording. Directly inactivating the gene is the surest way, because if we make some modification that actually only affects that one gene, and we know that if you make a deletion with a P element, which is what I'm going to describe in the next video, and the DMAP1 gene isn't actually there, that's loss of function, and that's for sure, right? Okay, and I just want to go quickly through this terminology because this is review, but it's helpful. So a loss of function mutation is when you either reduce or eliminate completely the function of a gene. So if you make an amorph mutation, that's the one we were aiming for with our deletion. If the DNA isn't there, you're not going to have any protein made at all. But also if you make mutation in the gene so that the protein doesn't do anything, it's completely inactive, that would also be an amorph. Hypomorph means less. Hypo means sort of under. And what that means is you still have some protein, but there's not as much of it as normal, so you have less of it. Or the protein is active, but it's not fully functional, so it works, but kind of slower than usual, and so on. Enough that you can see a phenotype, but it's not a complete loss of function. So when we say loss of function, we're talking about reduction or elimination of the function. Gain of function, we are probably not going to deal with very much in the class, but just for completeness, it's increase in function or it's a new function. So a hyper, think about being hyperactive, too much protein, or the protein goes around and keeps doing the job. Maybe it can't be inactivated or something. So there's too much of something. Or neomorph, the protein actually is altered so that it goes to the wrong place in the cell and it does something that it's not supposed to do. A common one that I may talk about in this class, yes, I think I will, is there's a chromosome rearrangement that causes a signaling protein that is involved with cell division to be kept permanently in the nucleus, where it keeps promoting cell division even though it's not really time for the cells to divide, and this promotes a certain kind of cancer called retinoblastoma. No, chronic myelogenous leukemia. Retinoblastoma is a different one. Anyway, just so that you can have this as background, this will help you think about and use the terms correctly in the screen. And I will record a little bit more, probably 2 more recordings to give a complete background.